This is lecture 3, and we'll be going into the characteristics of innate and adaptive immunity, more specifically the cells. So we're going to be transitioning into from innate immunity into adaptive immunity. A quick recap. So remember, in natural immunity or innate immunity, we have two different mechanisms. We have external and internal. And external is, remember, to try to keep these organisms from entering into our body. So we have our benign flora and our anatomic physiology, including our skin, mucous membranes, etc. Also we have internal, which is once they get through our external barriers, then we have internal mechanisms that will come into play and start to try to contain this potential infection. And that's usually inflammatory responses involved with that, and then it will progress on to the phagocytic and endocytic responses. And then cells will come into play, and this will be the crossover into adaptive immunity, which we'll be talking about in this lecture. The external defense, as I previously mentioned, is to keep the microbes from entering into our bodies. So that would include our skin, our mucus secretions and cilia, lysosomes, our acid pH of our stomach, urine, the vagina and the skin all have specific pHs to help deter microbial growth. And of course we have our benign or normal microbiota that also helps in that regards as well. Our internal defense, so once the microbe has penetrated our barriers, then it is the job of the internal defense mechanism to start to come into play. So we have inflammation, so remember the sequence of events, and also remember that we have two different types of inflammation. We have acute and chronic. Acute phase reactants, we kind of talked about complement and CRP, so those will come into play in this internal defense. And phagocytosis, remember chemotaxis, and opsonins. So opsonins, remember, is that they help enhance phagocytosis. During this lecture, we're going to be going over the cellular components of innate immunity and also the cellular components of the acquired immunity and their interaction with the immune process. So what do these cells actually do when they get triggered, and how do they get triggered to respond to a particular immune response? Also, we'll be talking about different characteristics of the T cells and B cells, and we're going to briefly outline the sequence of events that occur when these specific cells interact with a particular antigen. We're going to include B cells, T cells, and learn about antigen presenting cells, also known as APCs. I will mainly be calling these antigen presenting cells APCs throughout the rest of the course. And we'll also talk about a couple of different types of T cells, and we'll be able to explain and know the function of each of these different types of T cells. Before we start talking about cells and specific types of cells, we need to understand where these cells come from. So all of our immune system cells are derived from what we call pluripotent hematopoietic stem cells, or hematopoietic stem cells. All our white blood cells, leukocytes, that come from these hematopoietic stem cells provide either innate or specific adaptive immunity. Okay, so our innate is basically from our myeloid cell line. Okay, and these cells are what provide our first line of defense. So these myeloid cells include neutrophils, eosinophils, basophils, and or mast cells, monocytes, macrophages, and dendritic cells. So all these myeloid cells come from the same cell line and will serve as our innate immunity. Here again we have our couple of cells that are very important phagocytes. So we have our neutrophils, macrophages, dendritic cells, and natural killer cells. Dendritic cells are a very, very important APC or antigen presenting cell. We'll be going over each one of these cells in this lecture, and I'll kind of expound on all these different types of cells. But dendritic cells, a lot of people has never really heard of dendritic cells, and they are highly, highly important and are the presentation of antigens to other cells. So we'll talk about all of these in this lecture. Here is a table with our myeloid cell line white blood cells. You will need to know the phenotype or the name of the cell and their effector function. I would get to know these. This information will only help you throughout your career, so please get to know the effector function and the phenotypes of these cells. This is a diagram of where we can find our immune cells. So our neutrophils, eos, basos, monocytes, and natural killer cells can all be found in our bloodstream. Some of these cells will actually go through diapedesis and get into our tissues, our neutrophils. When eosinophils make that transition, they are then called tissue eosinophils. Monocytes will actually become macrophages when they make that transition. So after monocytes, usually they have around 70 hours-ish of life in the bloodstream. So right near that 70-hour mark, they will make the transition into the tissue and start to grow and become macrophages. Depending on where the macrophage is, will determine what the name of that macrophage is. So for instance, connective tissue, we'll call macrophages histiocytes. In the lungs, they'll be called alveolar macrophages, but no need to get into detail about that. For this course, we'll just consider macrophages as macrophages. Okay, so let's begin our journey. So the first one is neutrophils. They have several different names for neutrophils. We could call them polymorphonuclear neutrophils, polys, or PMNs. I will use all those names probably at one point or the other during this course. So just be familiar with the different name types of these neutrophils. Neutrophils are the most widely white blood cell in our circulating system. Remember that we talked about diapedesis, so half of our neutrophils will be circulating in our bloodstream and around half will be what we call in our marginating pool. They'll be stuck on the blood vessel walls, and they'll go through this what I like to call a shift change every 6 to 10 hours, and So the circulating neutrophils will adhere to the blood vessel walls, and the ones that are on the blood vessel walls will break loose and start to circulate through our blood system. They do this because if something does happen, if we get a splinter, for instance, then we have neutrophils right there at the location of a potential injury, and they can get there quicker. Remember, we consider these neutrophils are Marines, if you will, because they're first on the scene. So they'll be waiting to enter the tissue system either by at random or chemotaxins, waiting on those chemoattractants to seep into the blood vessel and to activate, say, hey, we need you in the tissue right now because something's wrong. So our neutrophils are the first ones to arrive, like I stated previously, and not to underestimate any other branch of military, but we consider these to be like our Marines. So they're usually the first to arrive and try to control the situation before it gets spread and out of control. So their main mechanism is phagocytosis. So that's what they do. That is one of their primary goals, is to phagocytize invading microbes. All right, the neutrophils are probably the best effective killer of microbes that we have in our body. The only limitations that neutrophils have are that they cannot they're not very effective against fungi and parasites, but we do have other cells that actually make up for that inadequacy. I will not spend too much time on this slide. Most of us has either taken hematology or is taking hematology right now, so basically neutrophils are multi-lobed cells with purple granules inside the cells, and these granules are basically what we call lysosomes. So these are the enzymes that help them kill a bacteria once the bacteria is engulfed. The next cell are our EOs. This is probably one of my favorite cells to see in a differential because I just think they look pretty awesome. So our EOs are probably less than 5% of our circulating white blood cells. They can phagocytize, but not as efficiently as our neutrophils. Our EOs have granules, heavy, they stain red granules. They are bilobed, and these granules are actually contain previously synthesized proteins, including like catalase, lysozymes, certain cytokines or chemical messengers, okay, growth factors, and cationic proteins. Although these eosinophils are capable of phagocytosis, they're just not as efficient as neutrophils because A, they are present in smaller numbers, and they lack digestive enzymes. However, one of the main functions of EOs are to A, neutralize basophil and mast cell products. So when we have an allergic reaction, basophils and mast cells start to release all this histamine and heparin, and EOs are designed to go up and clean up their mess. Also, it can kill some parasites. So these cationic proteins can actually help damage cell membranes and kill larger parasites that can't initially be phagocytes. So we're talking about like worms and etc. So yeah, so EOs play a major role in allergic reactions and parasitic infections. The next cell we'll be talking about is basophils. So this is a cell that is one of the least circulating cells in our bloodstream. Basophils have a very, very short life, usually only a couple of hours, and it is highly identifiable by the intense, big, deep purple granules in the cytoplasm. And now these granules contain histamine, cytokines, growth factors, and even small amounts of heparin in these granules. So when these basophils get activated, then the basophils will release all this histamine and heparin, which induces an allergic reaction, along with mast cells as well. We'll talk about those next. So when an IgE, an immunoglobulin, or IgE antibody binds to their cell wall, this is the steps in the basophils getting activated. The next cell is the mast cell. So mast cells can be found in different tissues throughout the body, mainly connective tissue, especially around certain blood vessels, or our blood vessels and our lymphatic vessels. These mast cells have a huge role in hypersensitivity, like allergic reactions, etc. So like basophils, they could also bind to IgE and or other anaphylotoxins to become activated, and this will actually start the release of the granules inside the cell. Like histamine is in the cell. Also, that's kind of unique in that these enzymes in a mast cell also include acid phosphatase, alkaline phosphatase, and other proteases. So that's pretty unique to this type of cell, and especially with a long life, either from 9 to 18 months they can survive in the tissue, so they really have a long lifespan. And also another important thing about the mast cell is that they can actually both enhance and suppress the adaptive immune system. They can enhance it by functioning as an APC. So that is one way they can enhance, and they can suppress it by obviously releasing large amounts of histamine into the system and creating this allergic response. The next cell we'll be talking about is the monocyte. So monos can be up to 12% of our circulating white blood cells in our circulatory system. They have very fine granules. Sometimes they'll have vacuoles inside these monos, and the granules are actually of two types. The first type contains peroxidase, acid phosphatase, and aerosulfatase. Basically, this would be indicating that these granules are similar to neutrophils. The second type of granule may contain beta-glucuronidase, lysosomes, and lipase, but no alkaline phosphatase. So these monocytes will stay in our circulation usually up to around 70 hours or less, and then they'll migrate into our tissues where they will become macrophage precursors. The next cell is the macrophage. So these are monocytes that actually move into the tissue to become macrophages. So remember previously when I mentioned depending on what tissue location they enter will determine what type of macrophage it will be. So if they move into connective tissue, then it would be considered a histiocyte, lung tissue, alveolar macrophage, etc. These macrophages have huge phagocytic capabilities. Okay, so they produce intracellular lysozymes to help in this phagocytic process. They also have antigen presenting capability, so they are considered an APC. They present antigens to mainly T helper cells or T helper lymphocytes. Different ways that they can be activated. They can come in contact with a direct antigen and become activated. They'll engulf it and become activated, and then they present that particular antigen to a T cell. Alright, or they can become activated in response to cytokines from another T cell. So they can become activated several different ways, and remember that depending on where they're located kind of ultimately depends on their name, but for this course we'll just refer to them as simply macrophages. Here are a couple of images of macrophages. These are really large cells, and you can see in the picture on the left that they have can have vacuoles, especially if they are phagocytizing bacteria. The one on the right, you can see almost has like a pseudomembrane hand. I hate to call it hand, but appendage, I guess, reaching out towards this one bacteria in this picture. So they have a lot of different capabilities, and they're really truly a great phagocytic cell. So monocytes and macrophages usually arrive later to the site of inflammation. When they do arrive, they typically will remove unwanted matter. This could include invading organisms, toxins that the organisms may have produced, or products of inflammation. So they're almost like the cleanup crew of the inflammatory process, but they are very active phagocytes as well. So they help clean up the residual mess of the inflammatory process. Now they could also, another benefit of them is they could also secrete pro-inflammatory cytokines as well to help the inflammation process. If they happen to be there, right there where the let's say the splinter enters, then they can produce pro-inflammatory cytokines. Besides eosinophils, dendritic cells are one of my favorite cells as well. They are very under-recognized in cell terminology or cell identification. These cells are very abundant near the skin and mucous membranes, and what's special about these dendritic cells is that they go around in their particular environment and sample everything. They are specifically looking for antigens that don't belong in our body. So they are sampling our cells, our host cells, the environment around them, looking for bacteria or specific antigens that doesn't belong in our bodies. And they are they have very specialized phagocytic properties as well. They are one of the best antigen presenting cells, or APCs, in the body. They can activate our T cells. They can actually dictate how T cells develop in response to certain antigens. So these dendritic cells are very important in our fight against invading microbes or antigens. So our neutrophils, eos, basos, and monos are part of our innate immunity. But one cell that's kind of the odd man out here is our natural killer cell, and it is also part of the innate immunity. So our natural killer cell is technically part of the innate immunity, but we also can consider that this is like a bridge into adaptive immunity. So be mindful of that. It's technically considered innate immunity, but we can consider it as a bridge that is linking the two types of immunity together. Also, the natural killer cell is part of the lymphoid cell line and not the myeloid cell line. So also keep that into put that into memory and consideration. Natural killer cells are unique in that they are basically the bridge from innate to adaptive immunity. So these natural killer cells can actually kill microbe-infected cells without using any antigen-specific receptors. So if a cell is virally infected, then it can go up to that particular cell, and usually these cells are showing a CD marker. We'll learn about CD markers here in the coming lectures, that is stating, hey, something's wrong with me, you need to take me out, basically. So this cell will then release perforin and will induce apoptosis of that particular cell. Also, natural killer cells can secrete cytokines to help activate macrophages. This basically enhances the macrophage's ability to phagocytize microbes. As the slide says, it actually can make them meaner. Alright, so more aggressive. One thing to not get confused about is that don't get confused between natural killer cells and then natural killer T cells. They are two different cells. So remember when we were talking about innate natural immunity that we consider this to be non-specific, okay? This is our non-specific immunity. So the receptors on these cells that are part of the natural immunity can recognize broad structures or primitive structures. They're not specific. They don't say, okay, I know this is E. coli and I'm going to kill it. No, they can recognize very broad antigens in order to be able to attack a specific antigen. All right, and these structures are called pattern recognition receptors. So these actually engage in trigger pathways that will promote inflammation. Now, these structures are tagged and or recognized by cell surface receptors, which will then trigger their immune response, and we call these toll-like receptors. So these pattern recognition receptors are the same thing as toll-like receptors. They are just broad, primitive receptors that will identify a broad range of different antigen structures. So our toll-like receptors are present especially on our monos, macrophages, and PMNs, and these cells are constantly on patrol looking for certain microorganisms that they can detect with their toll-like receptors. So we have 11 different toll-like receptors, or known, with limited diversity. So these toll-like receptors are what we call automatic detection of pathogen-associated microbial patterns, or PAMPs. All right. So these toll-like receptors are predisposed or predetermined when the cell is going through the maturation process after it's been stimulated from a stem cell. It's like, okay, you're going to become a neutrophil. Then it'll go through gene rearrangement, and these toll-like receptors will be formed at randomly. Okay, so these random, and each cell will have these random toll-like receptors, so the chance of coming in contact with a similar antigen that it's trying to detect becomes much greater. Alright, so this is what toll-like receptors are. They are very primitive, but this does give a degree of immunity to certain organisms or antigens. And these are, remember that these monos, macrophages, PMNs with the TLRs, or toll-like receptors, are part of our innate immunity. So that kind of helps tie in the primitive aspect of our innate immunity, and in conjunction with our myeloid cell line cells. So here's a quick review. If you'll list what cell you think goes in each box, we'll go over the answers in the next slide. So one of the most potent phagocytic cells in our tissue, it would be our dendritic cell. Natural killer cells kills infected cells without using antigen receptors. Macrophages enters tissue and specializes. Okay, for instance, microglial cells, alveolar macrophages, histiocytes, etc. PMNs, remember, is our first phagocytic cell to reach the site of infection. Our basophils are usually associated with hypersensitivity reactions, aka allergic reactions. Very short lifespan. Monocytes are an APC that actually circulates in our blood system before migrating into the tissue to become macrophages. Eosinophil is associated with allergies and parasitic infections, and mast cells can be found on mucous membranes and in our skin. So let's begin finally talking about the cells of the adaptive immunity, and these include our lymphocytes, our B cells, and our T cells. And we'll talk about more in depth about our antigen presenting cells and what role they play in our adaptive immunity. So antigen presenting cells, or APCs, can be either macrophages, dendritic cells, and even some B cells can be APCs as well. And all these antigen presenting cells are located in our skin, throughout our skin, our lymph nodes, spleen, thymus, our respiratory tract, GI tract, anywhere where there's a potential to come into contact with a foreign antigen. A lot of these, the main role of these APCs is obviously to present antigens to lymphocytes in order for them to become activated and to help prevent further infection in our bodies. Now, how do these APCs present these antigens? So they have these special molecules called a major histocompatibility complex, or MHC. And we'll speak a little bit about MHC in the following lectures. First off, do not let this slide overwhelm you. So this is a general representation of an antigen presenting cell presenting an antigen to a T cell. So when an antigen or an APC comes into contact with a specific antigen, it's going to typically phagocytize this antigen, break it up, and take a piece of this antigen to present it to a T cell or another lymphocyte. So in order for this to happen, the little piece of antigen must be will be put into an MHC, a major histocompatibility complex, okay, and then pushed out to the surface of the cell. And then a T cell will come along with, remember, the T cell receptor, or TCR, all right, and then it will bind together with the MHC, and that is what we consider the first signal. So if the TCR recognizes the antigen which the APC is presenting with the MHC, then that will trigger the second signal. So then the APC will deliver what we call a transducer to, and this will activate the T helper cell, the T cell. So don't get too overwhelmed. We'll be definitely going further into the actual process of the APC presenting this antigen in a later lecture. So I just kind of want to give you an overview of what's happening and what's going on when this APC presents this antigen to, for instance, a T cell. So let's take a look at our lymphocytes. So we have a lot of lymphocytes circulating in our bloodstream. Around 20 to 50% of our white blood cells circulating are lymphocytes. All right, and they have specificity, and they provide antigenic recognition. Okay, so we have T cells that has immunoregulation and cell-mediated immunity, and we have B cells that actually has antibody production capabilities and is part of the humoral immunity. One thing to keep in mind is that only cells with specific receptors for antigens are able to produce memory response. So I'll go ahead and give you a little foresight into this. So if it's, we'll talk a lot about this later in later lectures, but if the antigen is made up of a protein, then this will create the T cell or B cell to produce memory cells, but the antigen has to be a protein in order to do that. To go on with lymphocytes, all of these lymphocytes are produced in the bone marrow, or start out in the bone marrow. So what type of antigen receptors do these B cells and T cells have? Well, pretty easy actually. The B cells have B cell receptors and T cells have T cell receptors. So let's first take a look at our B cell. So our B cells are covered with antibodies. Okay, so these antibodies that are surrounding the B cell on the outside membrane is considered to be the B cell receptor. All right, and there's not just, how many is that? 3, 6, 8. There are hundreds of antibodies lining these B cells. So there are multiple ways that this B cell can potentially come into contact with the specific antigens that these antibodies can detect. Now all these, we'll go into this further later, but all these antibodies on this B cell is specific for only one type of antigen. Just keep that in mind. So we have our T cell receptors, or our TCR, and we consider this to be unique to T cells. So B cells have the antibodies, T cells have the TCR, or T cell receptor. So let's talk about our B lymphocytes, or our B cells. Remember, they are part of our humoral immunity, and our B cells are the only cells that are capable of producing antibodies. So when we are talking about B cells, sometimes we'll say a naive B cell, and basically what a naive B cell is, that it's a B cell that has not come into contact with its specific antigen that it's trying to detect. So it's naive. It has no previous contact with an antigen. Once it does come into contact with its specific antigen that it's made to detect, it will then become activated. It'll proliferate and differentiate into memory cells. Remember, if it's a protein antigen, into memory cells and effector cells. And for a B cell, the effector cell is called a plasma cell. And these plasma cells is what is actually secreting antibodies. So let's take a look at this diagram. So this is one thing to remember, is that remember our B cells, when they are made to detect one specific antigen. Alright, so one B cell can detect one specific antigen. It has what we call, or I like to call, a 1-to-1 ratio. So we have one B cell, one antigen. So when it finally comes into contact with that antigen, that B cell will become activated. It'll start to proliferate and differentiate into memory cells and plasma cells. Now, remember that if this antigen is of a protein variety, then this B cell will differentiate into memory cells, and those memory cells will have that exact same antibody coating the membrane as what first activated that B cell. So it will then differentiate into plasma cells as well, and when these plasma cells start to produce antibodies, those antibodies will be for that specific antigen that activated that B cell also. So we'll have millions and millions of antibodies being produced towards that specific antigen. So our T lymphocytes are part of our cell-mediated immunity, and all mature T cells will have T cell receptors, or TRCs. They are similar to the antibodies on our B cells in that they are very unique for a specific epitope, so they can only detect one type of antigen. It can recognize an antigen when it is presented with a MHC from the antigen presenting cell. So remember the major histocompatibility complex. It is presented, the antigen is presented on this MHC to the T cell, to the T cell receptor. We'll kind of briefly go over that in the next slide. So here is a crude drawing of the process. So we have our antigen presenting cell in the pink, or is that purple? My eyes are not what they used to be. And then we have our T cell in the bottom with our T cell receptor. At APC will have the major histocompatibility complex, and this peptide, or the antigen, the APC is presenting it to the T cell. So this T cell will lock up to the MHC, and if it detects, if it's the same antigen that it recognizes, then we have what we call a T cell surface glycoprotein that kind of latches on to the MHC, and that's like the second signal, and that's where the T cell will become activated once those two signals are reached. But we will go further into this, and I'll have other diagrams that will help explain this process. So our T lymphocytes are basically split up into a couple of different subsets, I guess you could call them. So we have our T helper cells and our cytotoxic T cells. Our T helper cells interact with APCs. When they become activated, they become effector cells, so they will start to secrete cytokines, or our chemical messengers, and these cytokines actually help activate other immune cells, including B cells, T helpers, other T helper cells, cytotoxic T cells, macrophages. So the T helper cell is actually a very, very beneficial cell in our immune process. We have to have this cell in order for it to communicate with other cells. So here's a quick diagram. So we have our, on the far left, we have our APC that has engulfed or phagocytized an antigen. It is going to process that antigen and then take a piece of that antigen and put it on its MHC molecule and present it to a T helper cell. And this T helper cell is going to recognize that piece of antigen and then start to activate and proliferate and differentiate. And it's also going to start releasing cytokines. So when this T helper cell is going to differentiate into other T helper cells, and then these T helper cells are considered to be effector cells since they have differentiated from the cell that was first stimulated. And these effector cells will start producing cytokines as well to help stimulate other immune cells to become activated. So the other T lymphocyte is the T cytotoxic cell, or the TC cell. This is heavily influenced by the T helper cell, so it's waiting on some cytokines from the T helper cell to help it stimulate itself. These cells tend to kill infected cells with viruses or microbes inside the cell. That, you know, there are some bacteria that actually, or parasites that infect cells. Viruses are one. Viruses actually use cells to help themselves replicate. So these cytotoxic T cells actually help kill those cells and to prevent the virus from spreading. Same thing with tumors. Tumor cells, the cytotoxic T cells help kill tumor cells as well. So our TC cells, or cytotoxic T cells, when they become activated by the cytokines produced by the T helper cell, they in turn become cytotoxic T lymphocytes, or CTLs, and these are tumor cell killing machines. Okay, when we have tumor cells in our body, we actually have a natural defense to these tumor cells before all the chemo and all the other methods of trying to kill tumors. Our body actually has a natural defense, and these are the CTLs. So here's a quick diagram of our B cells, T cells, and our CTLs. So it all begins with an antigen. Okay, either the B lymphocyte can phagocytize or come in contact with an antigen, engulf it. A macrophage can phagocytize an antigen and then present it to a T helper. or the antigen can infect the cell, and then a CTL can destroy that cell. But it all comes back to the T helper cell. T helper cell has to help activate the B cell and the CTL in order for this to occur. So the T helper cell is paramount in our immune response. So the T helper cell will release cytokines to help stimulate the B lymphocyte in order for it to proliferate and differentiate into memory cells and plasma cells. And it also releases cytokines to help the cytotoxic T cell to become a CTL in order for it to kill or lyse the infected cell. Here's a quick review over adaptive immunity if you want to fill in the blanks, and we'll go over the answers on the next slide. So the cells of the acquired immune response include T cells, B cells, and APCs, or antigen presenting cells. What are the two types of T cells? We have the T helper and the T cytotoxic. And the antigen receptor for the B cell is an antibody, and the antigen receptor for the T cell is the TCR, or T cell receptor. This is the end of Lecture 3. In Lecture 4, we'll be going over the lymphoid system.